Good day everyone, today we'll be discussing blotting methods. So, colony hybridization is the use to identify colonies that carry a gene or complementary DNA, cDNA of interest. So what is colony hybridization experiment? So a master plate containing bacterial colonies is used as starting material. Then each bacterial colony is composed of bacterial cells containing recombinant vector. Then the goal is to identify a colony that contains gene of interest. So cDNA or the complementary DNA, it is a DNA synthesized from a single-stranded RNA template interaction catalyzed by the enzyme reverse transcriptase. So then a nylon membrane is laid gently onto the master plate. Then after the membrane is lifted, some cells from each colony are attached to it. Then the nylon membrane is then treated with detergent which dissolves the bacterial cell membranes and make the DNA accessible. So the DNA within the cell is then fixed to a nylon membrane and denatured with sodium hydroxide. Then the membrane is then merged in a solution containing a probe which can be oligonucleotide, a fragment of DNA from a cloned gene or a specific RNA. The probe can be labeled with a dye for ease of detection. The probe is given time to hybridize to DNA on the membrane. The unbound probe is then washed away. If the DNA within bacterial colony did hybridize to the probe, a labeled spot will appear at the corresponding location. The pattern of the spots on the membrane is compared with the master plate to identify bacterial colonies carrying beta-globin gene. So the colony hybridization, so first colonies in the agar, then a nitrocellulose sheet, a blob blot, then the lyse bacteria denatured DNA, then identify the positive colonies, eco-compare. Colony hybridization. So Southern Blotting, so it is used to detect presence of a particular gene sequence with many mixture of chromosomal DNA. So it was developed by Edwin Southern in 1975. So it can determine the copy of the number of gene. Then it can also detect small gene deletions that cannot be distinguished under light microscope. So it can distinguish homologous members in a family. It can identify homologous genes in different species, zooblot or Jungferdose for animals. Then verification of transgenic organisms carrying new or modified genes. So it is important. If important variables in Southern blotting procedure are the temperature, ionic strength of hybridization, and wash steps. If the procedure is done at very high temperature or at low salt concentration, the probe DNA and chromosomal fragment must be very complementary in order to hybridize a condition called high stringency. So conditions of high stringency are used to detect close or exact complementarity between probe and chromosomal DNA fragment. So a transgenic organism is an organism that has DNA from another species incorporated into its genome via recombinant DNA techniques. So if naman the temperature is lower and or the salt concentration is higher, DNA sequences that are not perfectly complementary may hybridize to a probe, a condition called low stringency. So conditions of low stringency are used to detect homologous genes with DNA sequences that are similar but not identical to the cloned gene being used as a probe. So the technique of Southern blotting, so chromosomal DNA is isolated from the sample cells and then digested with restriction enzymes to yield many DNA fragments. Then the fragments are separated by a gel electrophoresis according to the molecular mass and denatured with the sodium hydroxide. So the DNA bands within the gel are transferred to a nylon membrane, a step known as blotting. Then the blot is then placed in a solution containing a labeled DNA probe. So the binding can be done under conditions of high or low stringency. So when hybridization is done at high stringency, the probe recognizes a single bond. But when hybridization is done at low stringency, this band and two additional bands are produced. Northern blotting naman used to identify a specific RNA and was originally termed as the reverse Southern blotting. It is used to investigate transcription of genes at a molecular level. So you can determine if a specific gene is transcribed in a particular cell type or at a particular stage of development. It can also reveal if a pre-mRNA transcript is alternatively spliced into two or more mRNAs of different sizes. Similar to Southern blotting with a few important differences. So RNA is the starting material and is extracted and purified from living cells. So this RNA can be isolated from a particular cell under a given set of conditions. Then after the RNA is extracted from cells and purified, it is loaded onto an agarose gel that separates the RNA transcripts according to their size. So the RNA within the gel are then blotted on a nylon membrane and probed with a labeled fragment of DNA. So RNAs that are complementary to probed DNA are detected as labeled. Western blotting method used to identify particular proteins with a mixture of many different protein molecules. This can be determined if a specific protein is made in a particular cell type or at a particular stage of development. So it's just the same with the southern and northern blotting, pero ang in-identify niya ay protein. So proteins are extracted and then it is loaded onto a gel and separate them by molecular mass. So to perform the separation step, the proteins are first dissolved in SDS or disodium dodecyl sulfate, a detergent that denatures proteins and coats them with negative charges. So the negatively charged proteins are then separated in a gel made of polyacrylamide, thus called as the S-page or the polyacrylamide gel electrophoresis. Then proteins within the gel are blotted into a nylon membrane, and antibodies is used as probe, which binds to sites known as the epitope. So primary antibody is given sufficient time to recognize the protein, and then the unbound primary antibody is washed away, and then the secondary antibody is added. So the secondary antibody binds specifically to a region in the primary antibody. They may be labeled or conjugated to an enzyme and are available commercially. In Western blot, they provide a way to detect proteins in a gel blot. So Western blot is used as a confirmatory test for HIV. So bands in gel are being evaluated against controls to determine the presence or absence of the infection. So this is the HIV Western blot banding pattern. So must markers. So what do you mean by the must markers? They must be seen. We have the P24 and the GP41. The good markers, the GP120 and the GP160. So at least one of the must markers should be present to be test positive. Absence of the must markers means a negative. So good markers may indicate that there is a developing infection but are not conclusive. So intermediate results should be retested after 6 months because 6 months it is the HIV window period. Western blot examples of the reaction of HIV. So positive control, we have the GP41 and the P24. So that's positive.